All right, welcome back. So now we're going to move on to this next unit, and I really love the second half of this course because it's all my favorite stuff. I love the cell cycle, I love talking about DNA, I think genetics is great. So today we're going to start with chapter 6. Chapter 6 pretty much talks about reproduction at the cellular level, just like the topic says right there. I'm going to break this up into several parts. This first lecture is going to talk about how chromosomes are organized and introduce what the cell cycle is. The next lecture is going to talk about cancer, because cancer is basically when the cell cycle goes awry, when problems happen and cells just keep dividing and dividing and dividing and eventually form a tumor. And then the last lecture we'll spend a little bit of time on is mitosis, or basically how the cell goes through the process of going from one cell to two. I spend a lot of time on the mitosis one, so when we get to the next chapter, which is meiosis, you'll see a lot of similarities and it goes a lot easier. So let's get into this one. So here's a lot of facts about the basics of chromosomes. So remember, chromosomes are basically stretches of DNA. Now, another term that I might have thrown at you before is the term chromatin. So this is DNA plus protein. And I'm going to show you in a few slides about how we organize this, because I think this is a really cool fact. Any cell in your body, except your gametes, if you go through, because they don't have half their chromosomes, but an eye cell, toe cell, skin cell, liver cell, if you pick just one random cell, and we took all the DNA out of it and stretched it out end to end, it'd be 6 feet long. I find that fascinating. I really do. 6 feet in one single cell of your body. So that is really, 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 really cool. Now, when I talk about chromosomes, so you look at this diagram here on the side, we're going to talk about how we organize it. So first off, we're going to start with the DNA. We talked about what DNA is. It's composed of the nucleotides, and we'll go in a little bit more of a review in the next unit. What we're going to do first is we're going to wrap these DNAs around what we call histones, which are protein subunits. And eventually it's going to look to like this right here, where it looks like we have a bunch of beads on a string. And then what we do next is we keep condensing and condensing and condensing and wrapping around more and more proteins to where we get to what you're probably used to seeing, that happy little X, which is a chromosome. Now here's some other cool facts about them. The number of chromosomes is going to be varies amongst different organisms. So just because you have more chromosomes doesn't make you more complex. Some key things about it, they're going to be fixed for a species. So this is going to prevent a lot of crossbreeding from happening. So no matter how much the dog wants to hump the cat, due to the differences in the number of chromosomes, maybe the dog has 48, the cat has 62. When things like in mitosis and meiosis or all the other stuff, the chromosomes don't match up, and the numbers are just too off, so then you're never going to get a cat dog. Now, if two species are super close, if you think about the donkey and the mule, I think one is 62 and the other one is 64, you can get a hybrid. Then that's the donkey and a horse. The hybrid is the mule, sorry. And that one will be sterile because it has that number of 63 chromosomes, and that makes a problem when it comes to meiosis. So as I mentioned, this is one way to prevent crossbreeding. So if you look on this diagram here, this chart, it just shows you a lot of variability. It talks about the diploid number, which has to do with the number of chromosomes, the number of genes, and then the size of the genome. So you can see, when it comes to our viruses and the bacteria, very small. And as we start to get more and more complex, you start to see, first off, the size of the genome gets larger. The number of genes. So the size of the genome and the number of genes don't actually correlate with each other. Sometimes for us, we have a lot of what we call junk DNA. 98% of our genome doesn't code for genes, so we just carry it around because we're horse pretty much. But you can see a lot of similarities, which is kind of cool. So look at the good old tomato right there. Look at the number of genes in the tomato compared to its size of its genome. It's really efficient compared to us, where we have a larger genome but less genes. Now, a few more things, and I talked about this a little bit. A majority of your DNA is going to be found in your nuclear chromosome, so in that nucleus. But there is a little bit of DNA found in your mitochondria, and I mentioned how thanks mostly from mom, but now we are showing some research showing that maybe dad is contributing to that. When it comes to how your DNA is normally organized in your cell. So this will relate to cell cycle when your cells are in interphase, which is the phase that they're going to spend most of their time in. They're going to be in what we call this uncondensed state. And I use this analogy like a plate of spaghetti. There's different noodles. They're all about the same whatever. But it's really hard to see where one ends and one begins. It's like a hot mess. A hot mess expresses what your cells normally look like. They don't normally condense into these tight chromosomes until they're getting ready to undergo mitosis or meiosis. Just a few more reminders. The prokaryotes, which have no nucleus, remember that, but they do have DNA. They have one circular chromosome, and they're going to condense it down to that nucleoid region. And us eukaryotes, we have linear chromosomes. So they're just like lines, like think of that little pasta noodle, spaghetti. And we're going to keep our chromosomes in our nucleus. So how do we look at them? One way you can do that is we can take some actively dividing cells, just take a sample, and what they do is they halt them in metaphase. They use a chemical to halt the cells from dividing, and they halt them in a certain phase. And then they arrange what we call a karyotype. So as I mentioned, your cells don't just magically have all your chromosomes line up in order to make it easier for us. So this is an example of a karyotype right here, and this computer program actually color-coded. Your chromosomes are colored like this. They color-coded it, and then they match them up by size and make sure you have all the different pairs. Now, karyotypes, we use them to spot problems. Genetic problems is a big one, and I'll show you some examples in a few slides. But if you look at this organization right here, this is a human karyotype. So we have 23 pairs of chromosomes right here. If you look at the size, the largest chromosomes will be first. So as we go down, you start to see the size decrease and decrease. Now we're going to have two different terms. We're going to have what we call autosomes. So this is any chromosome not involved in sex determination. So this is pairs 1 through 22. These are your autosomes. And then your sex chromosomes will always come last. These are going to have genes involved in sex determination. It's your X and your Y. Or if you're female, it's your X and your X. So total, you have 46, but you have 23 pairs. And we'll talk about what that means in a little bit. But what we use these karyotypes is to spot genetic problems, because chromosomes like to do funny things sometimes. So you can have problems where you have a deviation from the normal number. So you might have short one copy, or you might have an extra copy. Some examples, trisomy 21. This is an example of Down syndrome. This is where you have 3 copies of chromosome 21. There's quite a few that fall into the category of the sex chromosomes. Those who really love to have problems with non-disjunction, and you'll get multiple copies with that one. Happen quite a lot. You can also have structural changes. So this is where parts of the chromosome like break. Sometimes they can go to a different location. They can switch parts with each other. They can translocate. So if you look at this example right here, we have normal chromosome 9, chromosome 22. What they did was they had a break, and then they just switched with each other. You're like, well, you still got the gene, but it's in a different location. Well, there are certain genes that have to be on a certain chromosome, called Hox genes, and they turn on a certain time. So if they're showing up in the wrong place, it's time for that gene to be turned on. It's not there, and it can lead to problems. There are all kinds of things that can happen. I don't want to get too much down the rabbit hole, but this is actually kind of cool. Chromosomes do a lot of fun things. So here's some examples of karyotypes. You can see some problems. So as I mentioned, you can have 3 of chromosome 21. This is trisomy 21. There's Down syndrome. Here's an example of the problem with the sex chromosome. This is that Klinefelter's ones. We've got 2 X's and a Y. Normal sex chromosomes, you're either going to have 2 X's, and you'll be female, or if you're male, you'll have an X and a Y. Now, one thing that has been noticed, like as mentioned with us females, we made all of our eggs before birth. And one thing that has been shown in the research and everything is how the maternal age affects how some of these chromosome problems arrive in the youth. Now, what happens is as the female's age, more and more likely that there's going to be some problems happen, and it'll probably happen due to problems in the egg, and you might get some of these problems happen. They're looking at common risk for Down syndrome in the different semesters, and you start to see it increase as the woman gets older. Not scaring you, that biological clock's not ticking, but it's not a guaranteed happen, but there's just more of a chance it will. Okay, so the importance of your sex chromosomes. Now, remember that's that last pair right there. It's really important because sex chromosomes do have some essential genes that regulate the body. The X is super important. If you don't have an X, you're pretty much going to die. The won't go to term. But you can have problems when you have multiple X's, only one instead of two. This is just a table showing you some of the different examples that you can have, which is quite extensive when you look at it. What happens, you'll see problems that really have problems with fertility, because a lot of genes associated with fertility are going to be on the X and Y. And this is just if you're really fascinated. Now, why the sex chromosomes are super important. If you're going to be male, it all comes down to that one gene, which is the SRY gene. Now, this gene is on the Y chromosome, and what it does is determine, in early development, I think it's about week 6, between week 6 and 8, we're all kind of like gender neutral at that part. At that point, if you have the Y chromosome, the SRY gene is activated. Now, once the gene is activated, you are committed going down the path of becoming a male. If it's not activated, then you will go down the path of becoming a female. The way they found that was actually taking this gene and removing it. So they actually took some early embryo developed mice and removed the SRY gene. They developed as female, even though they had the Y chromosome. They flipped it, so they added SRY gene to a female that had 2 X's and developed as male. So here's like a link to the article. I think it's really cool. So we're learning all kinds of weird things with science, and I thought that was really cool that just one gene really determines which gender path we're going to go down. So this video will talk a little bit about it too, but I want to point out this little image right here. It's kind of interesting if you look at it. Now, if you look at the size of the X and Y chromosome, you see a huge difference. At one point in time, the Y is thought to derive from the X, and then as time it's gone through and shrunk and got smaller and smaller. And there has been some question, is it continuing to shrink? Will it eventually go away? Which makes you think, so if the Y chromosome goes away, there goes away the entire male population. So watch this video. They'll probably answer that question for you. It's really cool. But it makes you wonder, it's like, at one point in time, these were the same size. What caused all this shrinkage to happen? So we saw this picture before, but we're going to go through it a little bit more. So we're going to talk about how we organize our chromosomes. So at the simplest level, we have the DNA. We talked about it. So it's all those nucleotides joined together, form that sugar phosphate backbone, the complementary bases pair with each other. And then it twists to form that helix. Now the next level is we're going to have what we call histones, which are proteins. They form like a little protein ball. And what happens is the DNA just wraps around. It's about 1.65 times that wraps around a histone. And it kind of will look like what we call that beads on a string. So this micrograph right here actually shows you at a 10 nanometer fiber, which is shown right here, of actually DNA wrapped around those histones. So those histones are those little dots right there, and the DNA is wrapped around them. So this is actually an image of DNA. It's really cool. And then we're going to keep coiling and coiling until we get to what we call a 30 nanometer fiber. And then we'll form supercoils, and then we keep coiling until we get to our chromosome arms. And you're probably used to seeing those little X's anytime you think about chromosomes or the cell. Now, most of the time, when your DNA is not replicated, so most of your cells just hanging out in the interface, it's going to be in this linear chromosome. So remember, you only have 46 or your 23 pairs. So of your 46 chromosomes, mom gave you 23 and dad gave you 23. So each pair, one was from mom, one was from dad. Now each of those together is called a homologous pair. So if this is chromosome 1, maybe mom gives you purple, dad gives you green, all right? Now, what we're going to do is when we talk about the phases, or S phase especially, when we're getting ready to go through cell division, we have to copy our DNA because we want to make sure the two cells each have the same exact copy. So what we do is we go through and replicate our DNA. So when we do that in S phase, we like to keep the same copies together. So what we did is we copied this one, made an exact copy, and we're going to keep them together. And we keep them together, a little protein holding ball right there. Like, think of like a staple, it's temporary. We kind of staple them together, and then these will called sisters. So the sisters, it's still the same chromosome, they're exact copies held together by that centromere. And then this will be a homologous pair, and we'll talk about those again when we talk about meiosis. Now, the centromere doesn't always have to be in the center. Sometimes they can be up here towards the bottom. They can be at the very end. It's all kinds of different locations. A little bit more anatomy, the telomeres, which are the ends of your chromosomes. Now, there's not any actual genes here. It's a lot of repetitive DNA because every time your cell goes through and copies itself, or copies the chromosomes, we lose a little bit. It has to do with some problems with replication. That 5' 3' creates a little bit of problem. So every time a cell divides, the chromosomes get a little bit shorter. It's one of the things associated with aging. So here's a really cool video. I advise you to watch it. We would have watched it in class. And what it does is it talks about maybe we could slow down aging and get a better understanding of cancer by studying the telomere. So another cool video. That's why I said I really love this kind of material. There's a lot of cool stuff. So here's some terms that will show up again, haploid and diploid, and they'll really show up when we talk about your cells. So there's different types of cells. There's haploid cells and diploid cells. And it all has to come down with how many copies of the chromosome you have for each pair. So haploid means you only have one pair of each homologous pair. Okay, that seems kind of confusing. So remember, with humans, we normally have 46 chromosomes. But when we make your gametes, egg and sperm, we need to cut that number in half because the egg and sperm are going to fuse, and you're going to have 23 plus 23 to get the 46. So when the haploid cells, when we do the process of meiosis, you're either going to get your paternal or your maternal copy when you make that egg or sperm cell. So you're only going to have one copy. So if this is chromosome 1, 2, and 3, you only have one copy of each. Versus a diploid, you're going to have both sets of the homologous pair. So you're going to have both the maternal and paternal copy in the cell. So this cell will have all 46 chromosomes. So this one would have 23. This one had 46 if we were talking about human cells. Now the last part I want to introduce is the cell cycle. So the cell cycle is we kind of break it up into what the cell is doing in the different stages that is going to be. My mouth is not working. Think about, you kind of have like a cycle that you go through life. You have the infant stage, then you have the toddler, then you have a child stage, then you go through the teen stage and young adults, and then you're middle age, and you're senior citizen, same thing, just goes through the same thing. Now we're going to group it up into two big parts. The first part we're going to call is interphase. Right? So interphase, if you look at this graph, is pretty much all this gray part right here. And we're going to break interphase into several subparts too after that. So the first big division of the cell cycle is interphase. And then the second big part is what we call the mitotic phase. And this is where we're going to go through the stages of mitosis and also cytokinesis going into there to two cells. Now in interphase, boom, 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 showing right here, we're going to break it up into three subparts. We're going to have what we call G1, S phase, and G2. Now in G1, this is a brand new cell, just finished cell division, so now he just brand new to the world. This is where the cell is going to grow a lot. They're going to synthesize a lot of proteins. It'll probably spend a good amount of time hanging out in G1. Now, some cells, what they can decide to do is they can leave G1 and they can enter what's called like a GO. So, let me draw this off right here. there's like a little exit ramp right here, and he can go into what's called GO. It's kind of hard to draw that, but they can leave and go into what's GO. It's like a cell quiescent state. That means they can just like hang out there. Cells that do this, some of them, they permanently leave and stay in the state. They know that they're never going to go through mitosis. Your cardiac muscle nerve cells, they are permanently in GO. Some cells, they can go and hang out in GO for a while, and then they can go back into G1 whenever they're ready. So this G1 is just like a good growth stage, kind of prepping and everything. Now at a certain point, it's going to enter into S phase. Now in S phase is the synthesis phase where we're going to replicate our DNA. We're going to copy all of our chromosomes in this phase right here. Pretty much once you commit into S phase, you know that you're going to undergo mitosis. After S phase, we enter into G2, which is also signals for cell growth. And this is where we're going to prepare. We're going to make sure that everything is duplicated right. We're going to duplicate all our organelles, make sure we're big enough. This is pretty much like a good check stage. You know, if you're, I'm a planner, so like if we ever go on a trip, I got to check everything. Do we have all the stuff? Do you have all your clothes? Do we take care of all the stuff? The mail, the paper, blah, blah, blah. That's probably a lot that goes on in G2. It's prepping because next, it enters the mitotic phase, which is mitosis. Now, when it comes to amount of time spent in the different phases, about 90% of their life is in this in the inner phase. Depending on the type of cell, as mentioned, some of them permanently go this route right here. Now, throughout your cell cycle, we do have checkpoints, which are great because they kind of serve as like a, hey, let's check right here. Are we ready to go to the next stage? Maybe you're not quite ready. You need to hang out here a little bit longer. Now, there's three big ones that we'll talk about. This first one is a G1 checkpoint. So this point, if the G1 checkpoint, we're going to check before we go into the S phase. The purpose of this is to make sure the cell is big enough. If you're not quite there yet, you're not quite big enough to go through this, we'll tell you to stop. Maybe you have to go back to hanging out in GO for a while. You need to get a little bit bigger. You're not quite ready to copy your DNA. You don't have all the proper enzymes to do that. So sometimes this is the first checkpoint right here. Now, once you commit going into S phase, you are going to undergo mitosis. So this is another one. Maybe it's like identity check. Are you sure you're ready to divide? Because it's going to happen. Another major one is this G2 checkpoint. Now, the G2 checkpoint is where we're going to check for a lot of damage, especially on your chromosomes, because we copied all the DNA. That's a lot of DNA to copy. So we're going to check everything, copy, you know, did we make all the copies right? Everything match up? There's any problems? Do we have all our organelles duplicated? Are we completely prepared? This is that last checkpoint before you leave. So this is a super important one. The next checkpoint, and we'll talk about when we get to mitosis, is called the M checkpoint. So this is going to happen in metaphase. What we're going to do, it's going to be like a little tug test to see if a microtubule is attached to both of the sisters. And the reason that we do that is it's going to prevent the thing non-destruction happening. So when you saw multiple copies of certain chromosomes, like on trisomy 21, that's because this checkpoint failed. Now, this is your cell cycle. And we're going to link that to cancer in the next lecture because cancer is when this happened to overwrite these checkpoints. So I'm going to stop this one right here.